Today, FDA approvals in multiple myeloma and lung cancer, and regulatory advancements in kidney cancer and CLL. Hello and welcome to OncLive News Network. I'm Laura Jones. Acting 4 months ahead of schedule, the FDA granted an accelerated approval to the CD38 targeted monoclonal antibody Daratumumab as a monotherapy for patients with multiple myeloma following at least 3 prior therapies. The approval was based on data from 2 open-label clinical trials. In the Phase 2 MMY 2002 study, Daratumumab demonstrated a 65% one-year overall survival rate and a 29.2% objective response rate. The median progression-free survival was 3.7 months. In the Phase 1-2 Gen 501 study, the one-year overall survival rate was 77%. The objective response rate was 36%, and the median progression-free survival was 5.6 months. The FDA's decision makes Daratumumab the first monoclonal antibody approved for patients with multiple myeloma. This also follows a breakthrough therapy designation and a priority review, allowing for an approval based on objective response rate as a surrogate endpoint. A full indication for Daratumumab is contingent on confirmatory studies, which are currently ongoing. The FDA granted an accelerated approval to Osimertinib for patients with advanced EGFR T790M mutation-positive non-small cell lung cancer following progression on a prior EGFR tyrosine kinase inhibitor. The approval is 3 months ahead of schedule and the first for a treatment following a rebiopsy and molecular testing. The decision was based on data from 411 patients in 2 single-arm studies. In data provided by the FDA for the AURA study, the objective response rate with Osimertinib was 61% for those with EGFR T790M mutations. OncLive spoke with Dr. Pasiena of the Dana-Farber Cancer Institute about the findings from this pivotal study.
So this was a study, a sub-study of the Phase 1 clinical trial where patients were biopsied at baseline, which everyone was required to do for the trial, and then at day 15 of the trial. What was interesting learning from this was that in a significant proportion of patients, when you actually went to biopsy them at day 15, there was no tumor left or not a sufficient amount of tumor cells left to actually be able to do the analyses. However, in a subset of patients where that was possible, there was a biomarker modulation, meaning that you can show that pre-treatment there was phospho-EGFR present. That went down with a drug treatment, which is expected. Downstream signaling went down. PD-L1 expression went down, and there was a corresponding increase in immune cell infiltrates. So the biology of what was happening in the patients was what we had predicted from the preclinical models, that the biomarker modulation would be seen there. However, the most interesting part, or one of the more interesting parts of this was, as I mentioned, the fact that in a significant portion of patients you could not do this because they had such dramatic tumor responses. The protocol was amended to allow the biopsies to happen a little bit earlier, but even with that, the numbers were still a fair number of patients did not have evaluable tissue. If you looked at then the patients who were able to have biomarker analyses compared to the patients who didn't because they had such dramatic responses, the response rate was actually higher in those patients in whom you could not do the biomarker analysis compared to the ones who you could.
In the second trial, known as AURA-2, the objective response rate was 57%. Along with Osimertinib, the FDA also approved the COBAS EGFR mutation test as a companion diagnostic. A full indication for Osimertinib is contingent on findings from confirmatory studies. Also in lung cancer news, the FDA has requested additional clinical trial data to support a new drug application for Ruxolitinib as a potential therapy for pretreated patients with EGFR T790M mutation-positive non-small cell lung cancer. The NDA for Ruxolitinib was based on data from the ongoing Phase 1-2 TIGER-X trial. In an initial analysis of the data submitted to the FDA, patients with T790M mutant disease who received Ruxolitinib at the 500 milligram dose had an objective response rate of 60%. However, at a pre-planned 90-day meeting with the FDA, drug maker Clovis noted that response rates had changed, initiating the call for a more thorough review. According to the updated numbers, evaluable patients treated with the 500 milligram dose of ruxolitinib experienced a confirmed response rate of just 28%. In 170 patients treated with the 625 milligram dose, the confirmed response rate was 34%. The company noted that a majority of responses could not be confirmed at subsequent scans due to progression, which frequently manifested as brain metastases. The FDA assigned a priority review designation to the PD-1 inhibitor nivolumab as a treatment for patients with advanced renal cell carcinoma following prior anti-angiogenic therapy based on an extension in an overall survival in the CheckMate 025 trial. In the pivotal Phase 3 study, nivolumab reduced the risk of death by 27% versus everolimus. At a minimum follow-up of 14 months, the median overall survival was 25 months with nivolumab versus 19.6 months with everolimus. Median progression-free survival was 4.6 and 4.4 months in the nivolumab and everolimus arms, respectively. In CLL news, a supplemental new drug application has been submitted for ibrutinib for use in combination with bendamustine and rituximab to treat patients with relapsed or refractory chronic lymphocytic leukemia or small lymphocytic lymphoma. The application is based on findings from the Phase 3 Helios study. At a median follow-up of 17.2 months, progression-free survival with ibrutinib was not yet reached, as compared with 13.3 months for the control. Also in CLL, a Phase 2 study exploring the PI3K delta inhibitor idelalisib in combination with bendamustine and rituximab for patients with previously treated chronic lymphocytic leukemia has been stopped early following a positive interim analysis, according to a statement from drug maker Gilead Sciences. Following a recommendation from an independent data monitoring committee, the trial, labeled Study 115, was unblinded, allowing patients in the control arm to receive idelalisib. The recommendation to halt the study was based on a statistically significant extension in progression-free survival and overall survival experienced by patients in the idelalisib arm compared with the control. And that'll do it for today. Thanks so much for watching OncLive News Network. I'm Laura Jones. We'll see you next time.